Hey, this is Mr. Mitchell with another science video. Go ahead and write down the questions and we'll get to the answers in the video. Today's video is going to be about cells. Now we have kind of mentioned cells, and we'll mention cells a lot more even, but this is a little bit on how we first got to knowing about cells. And you can see a couple of names on the screen: Robert Hooke and Anton von Leeuwenhoek, who were two of the original people who figured out that all living things have these cells. And if you take a look right here, this is what cork looks like under a microscope. Now cork is, you may think that it doesn't come from a living thing, but cork actually comes from a kind of tree. So in the 1600s, when Hooke and Leeuwenhoek lived, is when Robert Hooke had his own microscope. He had made his own microscope, and we'll look at that in just a little bit. But he looked at cork, slice a sliver of cork underneath his microscope, and he saw something like this. And you can see these little bitty, he called them small rooms in there. And he knew that cork came from a kind of tree, so he could kind of take guesses that the living thing, the tree, was made up of these cells, these empty spaces. Now, von Leeuwenhoek was of a similar era. He was also in the 1600s. You'll find out a little bit his microscope looked remarkably different than Robert Hooke's. But he too had made a microscope. And his microscope, he looked at lots of things. He looked at water. He would scrape things from his teeth. He would scrape things from his gum and look at them under the microscope. And I Googled a picture of water under a microscope, and this is what I came up with. And from what it sounds like, Leeuwenhoek looked at water under his microscope, and he saw all these little bitty things that were moving around that turns out were unicellular organisms. We've discussed that unicellular means they have one cell. And here's an example of one right here. A unicellular organism that's just, they were just moving around. And you can imagine if you looked at water under a microscope and did not expect to see little critters in your water, then that would be quite interesting, maybe even shocking for von Leeuwenhoek. But anyway, he found these unicellular organisms not only in water, but from scraping from his gums and scraping from his teeth. And this was the 1600s, and most people, just the fact that there were cells was kind of strange, and it really took a while for people to figure out the commonalities between cells. By the time the cell theory actually came about, it was the 1800s. And in fact, if I'm remembering correctly, it's about 1830s or so. It was German scientists which ended up coming up with this cell theory. The cell theory is, first of all living things are made of cells. All living things. Plants, animals, protists, animals at the bottom of the ocean that are archaea, kingdom that is at the bottom of the ocean near the hydrothermal vents. Every one of them is made of cells, and of course they could be unicellular, have one cell, or multicellular, like almost all the other animals and plants. The second part of the cell theory is that the cell is the basic unit of life. It's the basic unit in that it is the small unit of life. Sure, you can have bunches of cells which make up muscles. Sure, you can have bunches of cells which make up the lungs. Sure, you can have bunches of cells that make up the heart or the stem of the plant. But when it gets down to the basics, when you go down to the smallest part, it is the cell that you will see in every single instance, no matter what the function happens to be. And thirdly, you may remember we talked about spontaneous generation and how it's common for people to think that you could get, for example, flies off that just generate from rotten meat. This is to repudiate that cells arise from pre-existing cells. In other words, if you have an animal, you can't just get an animal coming from dust or dirt or non-living things. You have to have something living to produce something living, whether it's trees, whether it's other plants, whether it's at the bottom of the ocean, whether it's animals or humans even. You have to have pre-existing cells, pre-existing life to continue the life. You can't have just a spontaneous generation like seemed to be a very common misconception at some point. Okay, let's take a look at another slide here. I have right here Robert Hooke's microscope, or at least a replica of Robert Hooke's microscope. And you can see this microscope looks very homemade, and it was. Robert Hooke made that. One that looked just like that. Have something here to hold the cork in his case. You have a big barrel type thing. You look in the eyepiece and it focuses in, probably had some kind of lens in here to look at the cork. So that's kind of what Robert Hooke's microscope looked like. Now this right here doesn't look a bit like a microscope, but this is Van Leeuwenhoek's microscope. Van Leeuwenhoek's microscope, he would have the specimen, whatever specimen he had right here, and I suspect with the water, he probably has some kind of vial or something that held it, and in here you had some kind of lens, and he looked at it from the other side. Now this lens was probably some kind of concave lens which would distort or make this a lot bigger. And you can see this is, of course, not Van Leeuwenhoek, but somebody demonstrating how you would use this contraption as a microscope. You would just look straight through from the other side through the lens that was in that hole, and you'd be able to see the microscopic, what's the word I'm looking for, recreation from the concave lens in there. Let's take a look at more about microscopes. Here's Leeuwenhoek's microscope, and you can see it looks exactly like the thing before. Lens here holds whatever it is there. Ancient microscope in 1700s looked a little bit like that. In fact, that's probably looks a little more primitive than Robert Hooke's microscope. Here you have a mirror here. Now most microscopes up until recent times looked a lot like this down at the bottom, and the top looked a little bit different. But what you have light that reflects off of this and goes straight up through whatever you're looking at. You have to have some kind of light source in almost every microscope up until the more recent electron microscopes. But you have to have some kind of light source. So no matter what room you were in, you could tilt this mirror to whatever light there was in the room, and that way you would have focused in light that would come up. This is called a light microscope. And literally, if you were in the dark, you could not look at this at all. And that provided a light source. Now, what we have now is called an electron microscope. Now, all schools, of course, do not have electron microscopes, but you get your source from an electron beam. I don't know if you can tell that or not, but the electrons are shot through the microscope, and that's what gives you the contrast on your on whatever you're looking at. And here are a couple of pictures of what you would see under an electron microscope. You can note down here that this is 500 times, it's magnified times 5,000, I should say, not 500. But you get quite a good image, and now that is an image, it's more of a 3D type image because the electrons will end up hitting that, and you'll be able to see what it looks like from an electron microscope. And you could even look in an electron microscope in the dark, and it kind of recreates the picture of what it actually looks like, and in kind of a digitalized image, I guess you might say. Well, anyway, I hope you've learned a little bit about the history of us finding out about cells.